We've been talking about DNA replication and using this depiction of the replication fork where you have one leading strand and one lagging strand, which make up the replication fork. But remember, at the other end of the replication bubble, you also have a leading strand and a lagging strand. So one strand of DNA can be the leading strand and the lagging strand depending on its orientation with respect to the origin of replication. Replication of the leading strand is less complex, and you might expect this to be faster. So a key question is, how can replication of the leading strand be coordinated with replication on the lagging strand at each replication fork? Recall that there is a DNA clamp associated with DNA polymerase that keeps the two strands together behind the DNA polymerase as it moves along the strand that it's copying. This clamp must be loaded onto DNA, and there is a loader protein complex for the DNA clamp. That loader complex serves to place clamps on both the leading strand and the lagging strand, and it holds them together. This results in coordination of the movement of the replication fork between the leading strand and the lagging strand. This clamp complex, which requires energy in most organisms for the action it does, prevents the leading strand replication from outstripping the pace of the lagging strand replication. So you get coordination of leading and lagging strands. But this coordination raises an interesting question. Why is replication only 5' to 3'? Why can't there just be 5' to 3' synthesis on one strand and 3' to 5' synthesis on the other strand, so that it could be easily coordinated? If we look at the replication machinery itself, all of these enzymes that are involved in this very complicated process are brought together to form a complex that is functionally a machine. It is a machine that copies DNA. So you have all the necessary enzymes, factors, everything that you need at the same place to carry out this process. And that was sort of implicit in some of the interactions that we discussed last time, such as between primase and helicase, between DNA clamp and polymerase. Bringing all of these together because they need to act at the same place at the same time would be very important. The overall mechanism is similar in eukaryotes to what we discussed, which is based on prokaryotic mechanisms. But there are more components. There are more subunits to the DNA polymerase, for example. But the fundamental processes that we talked about are similar.